See the diabetes. I have listed here primary assays and secondary assay for inflammation. I have touched only these two areas only for demonstration purposes. Now you do the first primary assays. Now with the primary assay, it's advisable that even in the in vitro test system, you keep some kind of positive control. That is essential. Any drug in that area you can keep. You may not give a serious note to that positive control because from the bioassay, you will come to know the potential of the activity through the concentration. Because when you have tested the crude extract, higher dose is active, but when you come down to the fraction after purification, lower dose would be effective. Just see the next slide. Look this carefully. This is a very important slide for those who are working in this area. You see plant A. I have not named the plant purposely. This is plant A. Now you have extracted with methanol. Second batch you have extracted with 50% water and methanol. Third lot you have extracted with 100% water. You have made 3 lots: water, hydroalcohol, and alcohol. Now you find that the activity lies in alcoholic extract. Glucose uptake activity 190%, 10 microgram per mil. So your activity should be in microgram. If it goes to the milligram, no activity. Forget about it. So microgram level activity in the in vitro test system. Now this has not shown activity or maybe very high dose. So we have rejected these. Coming to this, focus on this and come down to defat it. This fraction, when you got it, remove alcohol and the residue you shake with hexane. Remove the oily material, see the hexane activity, no activity in hexane. Come down to chloroform or then ethyl acetate or butanol, then come to the active phase. You see how activity goes down. Chloroform shows the maximum activity and you see the 192%, 10 nanogram. From microgram, you have come to the nanograms. Now you are interested to go into the further purification. Further purification when you did, you found this compound, this compound, this compound, this compound you found. Now this compound gives you activity up to the level of 10 picogram. You see from the nanogram, it comes to the picogram. So this fraction is giving activity. Then other secondary assay also show very significant regulation, significant upregulation, and then significant expression of the genes. You see, this means both primary and secondary assays, most potential compound. But the second one, this is in 10 nanogram. This is 10 nanogram. This is 10 nano. It means either you work on a single NCE, single compound, or you combine all the 4 and take that fraction and see the activity. Activity will be potentiated. Moreover, that will be acting on different targets and it may be more potent. So this is the way to make a phytopharmaceutical fractionation process. This is just in brief. Now, what are the steps which are involved in a drug development, whether it is NCE, single molecule, or it is phytopharmaceutical? First is a hit identification. Now, hit identification comes your extract when you are extracting and then fractionating, and you get the activity and you get a hit through the primary assays. Then go to the secondary assays, confirm this activity, and it becomes a lead. If it is very active and tertiary also gives you active, it becomes a lead material. Now from the lead, you have to go further for your advanced preclinical data generation. That will be regulatory pharmacology, toxicology, everything. Because don't invest money if in the tertiary assays your product fails, doesn't give activity. So because at every step you have to submit the dossier to the drug controller that I have reached up to this stage and now I want to go to the next stage. So if your product fails, then better to drop it at that stage. So then comes the safety pharmacology, regulatory, and then comes the clinical. Clinical trial is the last phase because it's a huge investment in clinical trials. We have to follow all the regulations. So this is the broad scheme for getting a first hit identification. When you get a positive and you don't get excited, it's just a hit. You have to go to the lead. Lead is confirmed, you get some sort of encouragement. Yes, I'm on the right path. Then go to the next stage. So step by step, you see how the progress is going on. Well, this single molecule, I have already narrated about this. The structural diversity, novelty, complexity of the single molecule has its own attraction. Now, some of the examples I'll give, which give you a brief idea what kind of plant material we have worked or we are working on and how we are moving towards phytopharmaceutical drug development. From CDRI, we had earlier lead compound of the Guwahati-IN. Now, we have dropped it because material is not available. You see, it is an oleogum resin coming from a tree. And tree takes about 12 years to grow when it produces the oleogum resin. And hardly you get, say, 250 grams of the oleo resin. So it's not economically viable. Although the drug was produced, Cipla did market this product. That was Guwahati-IN, glucosidone Z and E. It was a real phytopharmaceutical example of the product. But then the Cipla has to drop it because the material, genuine material is not available. Then it becomes too expensive to get the material and then proceed for glucosidones. Second is a Bacopa. Now, Bacopa was developed by CDRI initially for memory plus, CNS, memory enhancement. And it remained the market for quite some time. People have made money out of it, but that's not objective. The objective is to go to the refinement of the material, get into the clinical trials, and see what happens. Now, this is a potential drug. Bacopa is a potential drug for taking forward towards dementia. And that's a gray area. Those who are working on CNS system, this is one of the potential lead which is already available in the Bacopa as a nootropic drug. And of course, natural remedy worked for quite some time. And so then is the Picoiza curua. And then comes the IIIM, Salaki, I have given an example earlier. Then we have others for respiratory system, ICMR. Very interesting experience, my experience at ICMR, when we are working on this Vijay-SAR. You see, Vijay-SAR, from where we got the idea. At Gandhi Bandar shops, tumblers were sold. That tumbler was made of the heartwood of the stem of Tirocatha marsupium. And they were claiming that if you drink water, your diabetes will be taken care of. And from there, we started working on it. So ICMR was involved, AIIMS was involved, PGI Chandigarh was involved in clinical trials. We prepared, standardized everything we did. And Professor M.S. Ahuja was the head of the Department of Endocrinology, topmost diabetologist. And he was very keen that we should continue this work. We continued for 4 years, this project on Tirocatha marsupium, because drug was made under my supervision. My laboratory was making the drug, standardizing, giving all the quality parameters, and filling the capsules. Other clinicians objected to Professor Ahuja. Professor Ahuja said, leave it. There are better drugs available in the market. Why are you bothering? It's a huge investment we are making and nothing we are getting. He said, this is the drug of the future. At that time, he's no more now, but he said, this is the drug of the future. Why did he say? He said, I have compared it with tolbutamide, which is a very simple, a mild anti-diabetic drug. And it is comparable to tolbutamide. So in the coming times, pre-diabetic will be more predominant than diabetes. Therefore, as a pre-diabetic, it is an excellent drug of the future. At that time, he said. That's how ICMR again followed his advice and started working on another plant, which we are currently working only for pre-diabetes. That's pre-diabetes. We are working on a very common material, which you must be knowing methi, Trigonella fenugreecum, that we are currently working in partnership with industry, and that is going very well. The pre-scientific, preclinical data is on the way of generation, and we are sending for the toxicology studies, and then we'll start clinical trials. It will take about 2 years' time before we come out with the pre-diabetic drug. Anyway, this is ICMR's activities. Then we have, at DBT, we had an excellent experience collaborating Ranbaxy, DBT, and we found that one of the plants, Sesam pereri, showed activity against all the 4 serotypes of dengue, which is very rare. All the 4 serotypes to be sensitive to this drug. And this now has been taken by Sun Pharma. Sun Pharma has taken over, and so they are working on it to put into the botanical drugs in America and phytopharmaceutical drug in India.